Fine. So this is just a kind of overview of what I've just said in some sense in a bit more detail. So the question, more precise question than what was on the title slide that you can think of as being addressed in this talk is, can we think of cells as cognitive systems, right? And you have to define what is meant by a cognitive system. So we are realizing that cells have very sophisticated control mechanisms, which means that they are controlling, let's say, the expression levels of genes, concentrations of proteins. There's a lot of things in the internal environment of the cell are being controlled based on inputs being received from the external environment, right? And you can also think of a cell as a decision-making system, right? So given certain environmental cues, it is responding in certain ways. So think of bacterial chemotaxis. So you have bacteria, they move towards sources of nutrition in the environment, following a kind of gradient, right? So that's the simplest example. One of the best studied examples of this kind of decision making is, let's say, chemotaxis. But almost all kinds of cellular modeling can be thought of as trying to model some sort of decision making process. Even the cell cycle, right? The decision that has to be made to, at a particular point in time, switch from one phase to another, or go into the division phase, mitosis phase, and so on, right? All of those can be thought of as decisions that are being taken, and so in that sense, there's some kind of cognition going on. You could say some sort of decision making going on. And if you think about it, two of the key notions in cognitive systems. What are key properties that characterize a cognitive or intelligent system? Memory and learning are two of the key things. You may not agree that they are sufficient for cognition, but they do seem to be necessary in some sense to call something a cognitive system. It should have some sort of memory. It should be able to remember patterns or events in the past and make use of them, and it should be able to adapt itself, learn in some sense how to respond to different kinds of inputs. And so, if you think of some of the things cells are doing, there do seem to be analogies to these notions of memory and learning, and we'll see these spelled out in some more detail in the model I'll be talking about. But yeah, it's kind of intuitively makes sense that these things seem to be happening in cells. So the actual mechanisms that are there inside cells. So most of you have heard of these kinds of specific kinds of networks. So a lot of work in computational biology, systems biology, actually is on these things, right? So gene regulatory networks, protein interaction networks, signal transduction, metabolic pathways, right? These are all things that most of you are familiar with, because a lot of the research actually is happening on these, on modeling these specific subsystems inside the cell, right? But here we are trying to ask a slightly sort of broader question, which is, given all of these things that are going on, which are all playing a part in kind of the overall control and decision making that the cell is doing, how can we understand sort of how these different mechanisms fit into the cell viewed as a holistic system, which is controlling different parameters and taking decisions, and could ideas from AI and machine learning be relevant? So again, most of you, I'm sure, have some familiarity with machine learning, right? Anybody who has not heard of machine learning? Right? I'm sure all of you have heard of it. Most of you might be working on it in some ways, because it's very prominent nowadays in computational biology and also in a lot of other areas. So we are definitely already using many machine learning ideas, right? But here what we'll also try and do is to think about, so after all, machine learning ideas initially came up as a way of trying to mimic human learning, right? So humans are very good at learning from experience and learning to classify faces or other visual images, or learning to classify linguistic inputs, or all kinds of things, right? And now we are using machine learning to try and automate many of these tasks that humans are good at. So machine learning was initially motivated by trying to replicate cognition, right? And if it turns out that in fact some of these machine learning techniques, AI techniques, are useful now for modeling the cell, for capturing certain aspects of the cell's behavior, could it suggest that in fact the cell also can be thought of as a kind of cognitive system, right? So that's sort of the question we are asking. To answer that, you need to kind of dig deeper into the modeling you are doing, because very often these machine learning models are used kind of as black box, right? So you just want to map from some input to some output. You want to solve a classification problem or a regression problem. We use machine learning for that, without thinking about whether what's going on inside the machine learning model is replicating what actually happens in the real system, whether it's a biological system or another kind of real system. But here, obviously, that is really what we want to think about. What is the correspondence between a computational model and the real system that it is trying to mimic? Okay, so this is sort of an overview, quick overview slide on some of these specific terms that appeared in the first point here, in particular gene regulation. So mostly in this talk I will be looking at trying to model gene regulation, which is also possibly the most widely studied problem in computational systems biology. So what is that problem, right? Especially for those of you who may not be from a biology background, this recap may be useful. So cells are essentially bags of proteins, right? So everything inside a cell is essentially made of proteins. They are the building blocks of the cell. And as you know, genes are in some sense a recipe for proteins, right? So you have the genes of the DNA which are then transcribed into mRNA and translated into protein. So if you think of a cell as a combination of proteins which are interacting in various ways, carrying out various functions. Of course, the other pathways I mentioned, signal transduction and metabolic, they also involve proteins, right? So you have phosphorylation, you have enzymes and metabolic, all of these things are proteins. Right? So proteins are basically doing everything. And how do proteins get produced by this transcription translation process? So one of the fundamental things you would like to try and understand is how is the production of proteins inside the cell controlled? Right? And so this is just a very simple picture of kind of this transcription regulation sort of process, right? And again, this is not very visible, sorry for the low resolution here, right? But you don't have to worry about all the text. The basic idea here is just that you have a piece of DNA which is a gene which gets transcribed into mRNA, and that will then get translated into protein at some point. And the rate at which this is happening is controlled by, for example, the binding of other proteins here which are called transcription factors. So the basic idea to take away from this is just that there are kind of these regulatory networks, and so I'll be using the board as well a bit. I don't have kind of everything illustrated on the slides, but this is anyway an idea that most of you are familiar with. So the thought is that you have a bunch of genes, and you can think of the ways in which these genes interact in terms of transcription regulation as a sort of network where if we are trying to understand the rate of production of gene 3, it is affected by, let's say, the concentrations of genes 1 and 2. Or more properly speaking, it would be the concentration of the protein corresponding to this gene. Right? But I am kind of using gene and protein interchangeably here. Right? Roughly speaking, every gene produces a protein. So if this protein is present in a high concentration, it will activate this, right? So this kind of arrow means it's an activating relationship, and this flat arrow means it's an inhibitory relationship. So if this gene is high, it will tend to suppress the production of this gene. Right? So these kinds of relationships exist, and the kind of mechanism typically is something like this, right? That the G1 and G2, which are the transcription factors, by binding here in certain ways, they can either activate or inhibit the rate of transcription of the target gene. So this is the basic phenomenon we'll be trying to think about modeling here. Right? But of course, the problem, part of the problem here is that of scale, right? So there are various ways of modeling these. The simplest kinds of ideas are just using differential equation models, which is what we'll be looking at in some sense. But if you have thousands or even tens of thousands of genes, which is what you actually have in a real cell, then the scale becomes so large that it becomes very hard to deal with these models. So that's one of the problems that is there. But so this slide is kind of a summary of some of the things that we have been doing and others have been doing. So broadly speaking, what you want is given data about the concentration levels of proteins, let's say under a bunch of different conditions, right? So if I just had these 3 genes or proteins, again I'm using them interchangeably, so G1, G2, G3, and now I kind of measure the amount of these proteins inside the cell under a range of different conditions, right? So these conditions could be either just points in time series data, or they could be steady state measurements of the cell under different conditions. Let's say I'm subjecting the cell to a certain kind of environmental condition, I'm putting it in a solution with a certain pH, or I'm injecting some chemical or some drug into it, right? So I can do experiments under a range of different conditions, and under every condition I can measure the expression level of all of these genes, right? So typically this is done using microarrays, which most of you will have heard of, but there are also other techniques which people are now starting to use for some of this stuff, things like RNA sequencing, for example. Right, so we won't go into all of that. For our purposes, the thought is that for every gene, you have a bunch of measurements under different conditions. So this is the second point. So given this, so you don't know this, right? You just have this, and from this you want to recover or reconstruct or reverse engineer this thing, right? This is in some sense the basic problem in gene regulatory network inference, which, as I said, is a very widely studied problem in systems biology, that given this kind of data, you want to figure out the regulatory relationships. Right? So this can be thought of as learning the structure of a dynamical system given some input-output characteristics. So these are like input-output measurements, given a certain condition, and let's say so in order to model this kind of relation, you should be saying that in a condition where, let's say, G1 is high, then you would expect G3 to be high. So you would expect a positive correlation between G1 and G3 across these conditions. Similarly, between G2 and G3, you would expect a negative correlation. So you can think of, if you are modeling this particular gene, then these are the inputs and this is the output, right? So in that sense, I'm talking about an input-output kind of thing. And so in general, we have been looking at a range of approaches for modeling and learning, both we meaning both in sort of my group, but also we more broadly as in the whole community. But some of the specific things we have been looking at IIT-Delhi, Petri nets, differential equation models have been very widely used. We look at one version of them here. Markov networks, Bayesian networks have been looking at more recently. Right? So I won't talk about most of these things. If you are interested, feel free to let me know. I can tell you more. But for now, I'll talk about something slightly different. This is just to sort of set the scene for this. Okay, any questions or thoughts? Feel free to interrupt me at any point. Yeah.
The time duration, how do we take it? The sequence to C3?
Time duration.
Are these random or?
Yeah. So these could be either time series or steady state data, right? So both kinds of data are used. So if it's steady state, then it just means that you put the cell in a particular environment and you waited until it reached steady state and then measured the concentrations of these. If you are doing time series, then yeah, I guess it depends on kind of how frequently you are able to. So if it's using microarrays, you have some kind of resolution in practice, right? I mean, how frequently are you able to take these measurements? So yeah, so those are, I guess, experimental technicalities. I also don't know all the details there, right? But for our purposes, you are given some data which could either be time series or it could be just steady state under different conditions, and from that you are trying to come up with some structure for these relationships. Right? So this is the thought here. Now, so as I said, we will not really be talking about this problem directly, but this is related to what I want to talk about. So we'll see what that relation is. Any other?
Okay, so from what I'm getting is that if you do this measurement from a set of, for example, a person from India or any other species from Asia, and the other species could be from America or some other place, so will the results be, do you expect the results to be different?
So by species, so you mean if I do measurements in different species of animals or something like that?
Or different, the same species, for example, it could be Homo sapiens, humans, but it could be from Antarctica. So not Antarctica, but somebody living in North America, living in a different climate. So will the environmental factors affect the type of the gene, concentration or?
Yeah, sure. I mean, those factors can play a role, and you don't even have to go that far. Even within one person, there are so many different kinds of cells, right? So this sort of data will be very specific to the kind of cell you are looking at, right? So if you look at a neuron, it's very different from, let's say, a skin cell or a liver cell or heart cell. Right? So all of these cells are very different, and the reason they are different is because the gene expressions are different. Right? Every cell has the same DNA in it, right? The same 23 chromosomes are there in every cell. But the reason why cells are so different is because.
But if you compare the same cell to different people or two same species but living in different climates.
Same type of cell, you mean, for two different people?
Like for example, it's a neuron.
Yeah.
So if I compare neuron from somebody over here and somebody living in the northeast.
It could be different, right? I mean, so again, I don't think people have tried to look at. I mean, the variations even within, and there's a lot of noise as well in this kind of thing. I mean, gene expression itself inherently has a fair amount of randomness. In fact, we'll see one of the reasons why that might be. But yeah, so even if you measure the expression in different cells for the same person, or at different times for the same person or the same organism, you will see differences, right? And so definitely, if you are measuring different people, there will be many differences that could be there just because of some intrinsic factors and things like. So what you are getting at more is the external climate or something that could also have an effect, right? But I mean, it's hard to model the effect of external climate, I guess, on like individual cells because they're quite far apart, right, in terms of level of abstraction. So how is the external climate interacting with a hard cell or something? There would be, but it's quite a it's quite a distant interaction in some sense, right? Because there are much higher level systems in the body that are regulating body temperature and interacting with the environment. Right? So what you're asking is kind of a hard thing in the sense that it's very hard to try and understand fluctuations in gene expression for that kind of high level thing, right? But even if we just think about much more low level things in some sense, so here the measurements are happening just in terms of very local environment of a single cell, right? So that cell is placed in a certain solution which has certain pH, it has certain concentration of chemicals, whatever, temperature, right? All of these things you are varying in the local solution in which the cell is, and based on that you are doing these things. Right? So this is easier to kind of understand rather than something which affects the whole organism rather than individual cells. So that's the level at which people have normally thought about it. Right? Any other thoughts? Okay, so this is just a quick overview. Yeah, so this is one part of the picture which is just trying to say that we can understand. So this picture is saying we can understand fluctuations in the expression level of a gene if we know what are the other genes controlling, right? So ideally the situation would be that if I have this complete model and you give me G1 and G2, I can predict G3, right? And I can tell you based on the levels of these two what the level of this is going to be to a high degree of precision, right? But actually it doesn't turn out. I mean, all modeling that people have done actually falls far short of being able to do that. And one of the reasons is that there are many other things also controlling gene expression apart from just transcription factors, which are other proteins. And one of the things that has increasingly become of interest in recent years is non-coding RNA, right? So non-coding RNA are RNA that are produced from DNA that doesn't DNA that is not part of genes, right? So you have genes, as we saw on that picture, right, previous slide. So you have genes which are strips of RNA that get transcribed into mRNA. But in the whole sort of genome, which is 3 billion base pairs, the fraction that consists of genes is very small. Right? Vast majority of it is not genes. It is what has sometimes been called jump DNA. Right? People used to think that this DNA, because it is not coding for proteins, is not useful. Right? But memory there is an address, and to look up something in the memory you have to point out the address. So this is traditional memory, which involves a mapping from address to content, right? But for cognitive systems, although clearly, I mean, all of us have memory. I mean, we also use the word. Of course, the word memory originally refers to human memory, right? It only started being used for RAM later on, right? But the original sense of memory is the fact that we can remember things. But is our memory similar to, let's say, computer memory in this sense of being a kind of unique address to content mapping? But it seems that a more appropriate model for cognitive systems is what is called associative memory, right? And so here, rather than kind of having some unique arbitrary address, and if you think of your memory like that, you would say, well, what is that address? I mean, for the RAM, we can understand that there is a physical layout, and every location has a physical address, and of course that address kind of is encoded using a certain number of bits and so on, right? But for the brain, there doesn't seem to be anything like that, right? And what seems to be happening is that rather than having this kind of arbitrary address, most of the time you are doing association, right? So you are trying to retrieve something from memory, and the way that is retrieved is that a portion of that thing is presented to you, and based on that you retrieve the rest of it, right? So this is what associative memory fundamentally means, and another term for it is content addressable memory, right? So that means the address is the same as the content, right, or a part of the content. So you have associations between different things, and you are presented with part of that, and you retrieve the rest, right? This is how human memory largely seems to work, right? So here are a couple of examples, right? So can you fill in the blank? To be or not to be, that is. Right? So most of you can do this, right? Where is this from? Anyone knows the source? Right. Hamlet, right? So this is a very famous line from Hamlet, right? Similarly, this one, Concord, right? Where is that from? I think so, yeah, that's what I also remember. Not 100% sure. It's attributed definitely to Julius Caesar. Some of these can be mythical. So these are two very well-known examples of quotes that pretty much everybody knows as parts of popular culture, right? At least English-speaking people know them. And so by presenting a part of the content, you immediately are able to retrieve the rest of the content. So it's in your memory, and the best way to retrieve it, or perhaps the only way to retrieve it, is to give you a part of the content itself, and then you get the rest of it. More generally, you can think of one particular kind of instance of memory triggering, which I'm sure all of us have experienced at different points, is when some very small kind of particular stimulus triggers a much larger set of memory, right? So you might associate a particular odor with a certain place, right? Maybe the odor of a certain kind of food you associate with your grandparents' house or something, because every time you went there in childhood you got to eat that thing, right? So when you smell that thing somewhere else, you are immediately reminded of your grandparents' home, right? That kind of thing. Or sounds, right? So maybe there's a particular song you have come to associate with a particular place, because every time you went there that song is playing. So now when you hear that song somewhere else, you are immediately reminded of that place, right? So automatically, in a kind of involuntary fashion, this retrieval happens, right? So part of the associative pattern is presented to you, and the rest of it gets retrieved from memory. So these are simple examples. So this is what we mean by associative memory at the cognitive level. Now this talk is not about cognitive science. We are not trying to model human memory here. But the thought is that human memory is associative to a large extent, and there have been models that have been built to try and capture associative memory, which are called associative memory neural networks. We will be trying to use these models not for human memory but for cellular memory. So, and hence the title of the talk, or the theme of the talk, do cells kind of also do a kind of cognition? Okay. So is the idea of associative memory clear? It's a straightforward kind of thought. But let's see now how this is captured in neural networks. So before coming to associative neural networks in particular, let me say a little bit about neural networks in general. Right, so this part of the talk should perhaps be the most familiar to a lot of you, because you are mostly from engineering background. You would have studied neural networks probably at some stage. Right, so but I'm not going into any of really the mathematical detail, but just at a high level. So neural networks are a very widely used machine learning technique. They were originally inspired by trying to model how actual neurons work, right? Hence the name. So McCulloch and Pitts in 1943 came up with a mathematical model of a neuron, where the idea was that basically a neuron is something a bit like a switch, right? So the kind of activation potential of a neuron typically falls into one of two bins, right? I mean, it's either high or low. So you can, roughly speaking, think of it as a switch which is either on or off. And then the activation potential depends on some sort of integration of the inputs coming into it, which are coming from the dendrites that a neuron has. So it's sort of integrating electrical signals coming from other neurons and then applying some sort of threshold on top of that, and either it is on or off. Okay, so so this is a summary of the idea. Take a weighted sum of a bunch of inputs and apply some threshold to decide whether to fire or not. Okay. So here are some simple examples of the kinds of things you can do with this sort of model, right? And one reason why this has been interesting right from the start is because people were able to see immediately that this kind of thing can start to model basic logical operation. Right? So when you are starting to think of the brain as some kind of computer, can you model what the brain does in a similar way to what is happening on computers? So the realization that neurons may be able to simulate logic gates, which as we know are fundamental aspects of computers, was an interesting thing, right? So this is basically just showing you that. Right? So look at this picture. So each of these circles here is like a neuron. Like think of it as a neuron, artificial neuron, which is a switch, right? So that means these are all variables which are either 1 or 0, right? Now what is happening is these inputs are getting multiplied by the respective weights on these edges, 0.4 and 0.4, and getting integrated here. And then there is a threshold being applied here to decide whether this should fire or not, right? So let me just write it down slightly more formally here. I think it might help. So we have a picture like this. Let me call this here W1 and W2, for example. Let's say this is X1, this is X2, right? So these are these two neurons which are either 0 or 1. And this is my output neuron, let me call it Y. Okay? So the function that is being computed here is basically this, that Y is equal to, so let me write it like this, Y is equal to 1 if, so you are multiplying each weight with the corresponding input, right? So W1 X1 plus W2 X2. Right? So this is a weighted sum of the inputs being taken, and then this is being compared to a threshold, right? So over here there is a threshold, let me call it tau, for example. So if this thing is greater than tau, then it is 1, and it is 0 otherwise. Right? So this is the simplest kind of model of a neuron, which is basically what is being shown here, right? So in that picture, the particular values are 0.4, 0.4 and 0.5, right? But here I have drawn a more general picture. So you can see, why is it AND? Because if you consider all the possible values of the inputs, so if x1 and x2 are binary, there are only 4 possibilities, 0, 0, 0, 1, 1, 0 and 1, 1. And you just calculate y for all 4 cases. So if x1 and x2 are both 0, then of course this is 0, which is less than tau, which is in that example 0.5, so it will be 0. If one of them is 1 and the other is 0, then it will be 0.4, right? 0.4 into 1 plus 0.4 into 0 is 0.4, which is still less than 0.5. So both of these will also be 0. But if both of them are 1, it will be 0.4 plus 0.4, which is 0.8, which is greater than 0.5, so it will be 1. So you can see that this is the truth table for an AND gate. So with these particular settings, it is simulating AND. But the interesting thing is that by changing these variables, the same model can simulate OR as well, right? So it's a more powerful, more general model than just one logic gate. So just by making it 0.6 instead of 0.4, it now becomes an OR gate. And for NOT, of course, you only have one input, so you can easily simulate NOT by putting some negative weight here. So if this is 1, this becomes negative. If this is 0, it is 0, and then you can get by having a 0 threshold, you can get the inverse of the input at the output. It's a basic logical operation can be encoded by these kinds of neuron models, which basically come from the McCulloch-Pitts kind of idea, which was just the basic idea is encapsulated in this picture. Right? If you are taking some weighted sum, you are applying a threshold, and you are deciding whether or not to fire. Now, the last thing here is a slightly more general instance of how you can use these models. So this is where you just have a couple of binary inputs and you are using it to compute a logical function. But in general, what you could do is you could have a number of different features or input variables that you are measuring, let's say, of some kind of stimulus. and you are taking some weighted combination of that. So again, the numbers alongside the edges are weights, and then calculating some output entropy. So in fact, this network is meant to represent what can you guess what is this supposed to be, just by looking at the names there? Any thoughts? What kind of model do you think this is? Any thoughts? I guess it's hard to guess, without any context. But basically, so this is taken from this book by Steven Pinker, by the way, which is an interesting book to look at. So this is supposed to be a vegetable classifier. So basically, it's just a simple example of a model where, given any object, you are looking at a bunch of properties. Is that object green? Is it leafy? Is it crunchy? All of these have positive weights, and then it shows some other properties like magnetic, mobile, which have negative weights, right? So if you have a vegetable, for example, it will tend to have these properties and not those, right? So the weighted sum here will be a positive number. Imagine that the threshold is 0. Right? In fact, I have shown here 0.5. Depending on the kind of model you are using, typically there is a default threshold of either 0.5 or 0, right? So whatever that is. But you can see that vegetables, for example, will tend to have all of these on and those ones off. But some other object, like let's say, anything which can move, so actual, let's say animals, for example, right? So mobile here means a thing can move, right? So vegetables cannot move, but animals can move, or something that is magnetic. Or obviously they are showing a small number. You can imagine a bunch of properties. So things which are typically not associated with vegetables will cause the number to become negative. It will be multiplied negative, and so will it decrease the chances of getting a vegetable out. So this picture is just to show that one way you can use neural networks, which in fact is the way they are used in machine learning predominantly, is that you have a bunch of different features and you want to use some kind of combination of those features to get an output label of some kind, right? Saying that this input object is or is not a vegetable. You could use it for face recognition, is or is not a face, right? That is one, just to give one example of an area where it's being used very prominently these days, right? So if you look at, even Facebook now does kind of face tagging, right? So sitting underneath that is something like this. It's this kind of model, which is taking a bunch of features extracted from the image of a face and trying to give different weights to those features and finally arrive at a decision, is this a face or not, right? So this is a more general picture of how these kinds of basic neuron models can be useful. So they can encode abstract logical operations and also concepts and categories, like this is encoding the concept or category of a vegetable. Okay, so far we have looked at the idea that you can have a model like this and you can set all of these weights appropriately and get it to, yeah.
So, like as we are modeling these kind of systems using neuron, the concept of neuron that might be activated or deactivated, right?
Yeah.
And there is an interconnection of neurons also. So, this is easy to understand that something can be activated or deactivated, as in the sense of neuron. But when we consider this weight, connection between a neuron to another neuron, what do you think that there can be a mapping, the actual mapping to the human mind? Is there a distance between neurons or something like that?
The weight on these edges. You are saying what is the physical kind of correlate of these weights? Yeah, so actually there is, so the McCulloch-Pitts model was inspired by trying to model actual neurons, right? So there is a physical correlate to these weights, which is basically the strength of the synapse. So connecting any two neurons, there is a synapse, right, which is the place at which the axon from one neuron meets the dendrite from another neuron. So there is a synaptic strength, basically, which controls sort of how strongly the value of this previous neuron is being transmitted to the next neuron. Right? So roughly speaking, the synaptic strength is what this is thought to represent in the real brain. Right? But of course, it may not be exactly true, or there may be other factors affecting synaptic strength and so on. So that is somewhat speculative. Right? So you can think of this for now as just being an abstract model. It's not necessarily capturing every feature of how the actual neuron works, but it's inspired by that.
So this threshold is also like some function dependent or something like that, because different things, learning some other skills or require maybe some different threshold, or some movement may require some different thresholds. So there's some kind of functions of these thresholds, because these also play a major role in it.
Yeah. So it's a good point. So in fact, I am coming to that. So see, this model has 3 parameters, right? W1, W2, and tau. These are all parameters of the model, which in this case are set to 0.4, 0.4, 0.5. For or they are something else. For not, there might be something else. And for different things, as you are saying, we want different values for these. So we have set up this basic model, but we have not given you any way to actually set these parameters. I mean, if you know what the answer should be, you can set it. But supposing I just have, so for example, look at this one, right? I mean, I'm given all of these, but if I'm not given these, it's not easy to know how to set them, right? You would like some way to automatically set these given, let's say, a bunch of examples, right? So I give you a bunch of vegetables and I give you a bunch of other things, and you measure all their properties, and then you try to learn from those objects the appropriate mapping, saying that when green occurs, it tends to be a vegetable, so I'll put a positive weight here. When magnetic occurs, it tends not to be a vegetable, so I'll put a negative weight. So that is basically what learning is, right? Machine learning consists of taking a bunch of labeled examples. And so right now we have not described a machine learning model, we have just described a neuron model. The next slide will convert it into a machine learning model. So we're just coming to that, so that will address this question. There was another question.
So I was confused in the sense that this doesn't happen in the brain like this and or and not gates thing. It doesn't happen in our brain like this, right? I mean, because what I learned was that when we tend to do something, like if I tend to lift my hand, then before lifting my hand, the brain is going to send a signal and neurons are going to fire. So like according to your model, this thing, there should be some value attached to it, and if you do some and or not, if it reaches to some sort of a threshold value, then the neurons are going to fire and then I'm going to lift my hand. Does this happen for real, or is this just for mapping, like you told about the Facebook thing? Like if you see a face, if it has eyes, so it will have like 0.2 value. If it has a nose, 0.2 value. If it combines some of the values, and if it's more than 0.6, then it's going to be a face. So like this thing can be applied in machine learning, but it cannot be applied in, like, it doesn't happen in the real human body or near real.
Well, so we don't know, right? I mean, so again, obviously this is a very, very simplified model of what McCulloch and Pitts thought in 43. Of course, there have been various refinements, but still, I mean, this is a model which is trying to capture some very basic features of what neurons might be doing, right? And again, only individual neurons. So what you are talking about, let's say, are more complicated motor tasks, moving your hand and so on. So they would involve a lot of things, right? But you can imagine that you might be able to build up a complicated function by taking combinations of simpler functions, which may be things like and or not, right? Or if you think of your face recognition example, so you can see how this might model face recognition by Facebook, right? But can it not also perhaps model how I recognize that this is a face and that is not a face? I mean, so in my visual system also something similar must be happening. I must be integrating a bunch of features of what I'm seeing through my eyes, and then coming to a decision that this is a face and that is a chair and that is a laptop. So it's not completely implausible.
How can you find out the difference between two faces?
No, so of course you have many levels of this happening. We are not talking about that. And recognizing specific faces is something which still machine learning models are not very good at compared to what of course humans can do. But the point is that this is one set of ideas that could build up into such a model. We certainly don't have complete explanation or anywhere close to it for things that the brain is doing. Okay, so let's come to the learning part. By when am I supposed to finish?
You have some time, but the kind of expected time is around 1 hour, so we have another 55 minutes.
Okay, so I'll speed up a bit. I've been trying to go slower on this, but yeah, we can skip some of the details, right? So this is just to show that the next idea was this kind of perceptron idea, which, as I said, combined the neuron model with the idea of machine learning, and basically it said that you can take a bunch of labeled examples, right? So a bunch of objects which are vegetables and are not vegetables, and learn the weights by essentially setting initially all of these parameters to some random values, let's say, inputting the thing and comparing the desired output with the output you are getting, right? So you will get some random output if you have set these two random things, and it may be wrong, right? So if you want this to be an AND gate, you input 0, 0, you might get 1, you want it to be 0. So in whichever cases the output is wrong, you then adjust the weights, right? So if the output is 1 and you wanted it to be 0, it means you should reduce the weights. If the other way around, you should increase the weights, that sort of thing, right? So basically you can set up an algorithm which does that, and the perceptron is an example of a model which does it. Now one of the problems that this kind of model runs into is what is called the XOR problem, which is that it can model functions like AND and OR, but if you have a sort of a non-linear combination of the inputs, right, which means, so the simplest example of that is the XOR function, which is why it's called the XOR problem. If you have this function, this is a non-linear function because of the fact that the effects of x1 and x2 are not additive, right? So x1 and x2 individually cause y to be up-regulated, if you think of it as a gene, or increase activation. But when you combine their effects, this becomes 0. So this is a non-linear effect, and this cannot be captured by this, because if you have a high weight here and a high weight here to capture these two, then of course when you add these two, this will be even higher than that, right? This function will be higher for this case always than for these cases. So it cannot be that this is 0 when these two are 1. So this model cannot capture this, and so that was one of the problems with this, which meant that people thought this is a very limited model. But there was a simple solution, which is that rather than having a direct input-output mapping, you have some kind of intermediate layer, which is called a hidden layer or internal representation. And this is just a picture showing how you can solve the XOR problem by using a hidden layer. So now so each of these is the same kind of unit, right? So this into 0.6 plus this into 0.6, 0.5. This is actually an OR unit, right? The same thing from the previous slide. This is an AND unit. But now you are taking the outputs of these OR and 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 then feeding them to yet another neuron, which is weighting the OR with 0.6 and the AND with minus 0.2. So you can see that what this is doing is that if OR is true but AND is not true, then it will have a high value. But if both OR and AND are true, then this will push down the value. So that effectively gets you to calculate the XOR function which I've written there. Right? So this idea of being able to use a hidden layer makes the model much more powerful, because now you can capture non-linear effects. And these kinds of models are called multi-layer perceptrons, or more generally just neural networks. So the word neural network, as used in machine learning, basically means a model like this which has a hidden layer of representation, or more than one hidden layer as well. And as we'll see right at the end of the talk, the term deep learning, which is very popular these days, also refers to the same kind of model. The word deep just means that you have many hidden layers, not just one. Right? Otherwise, it's exactly the same kind of model. Okay, so the final sort of piece of the puzzle for understanding this idea of associative memory. So I've described basic neural networks. Now, if you look at this picture, so this part of it is the same as, for example, that vegetable picture. You have a bunch of properties, green, leafy, crunchy, and so on, which are feeding into an output unit that is calculating is it a vegetable or not. But what if you connect up all these units amongst themselves also, right? And now you try to learn weights here, saying that, so basically correlations. If this is high, then what is the chance that this is high or this is high? So if you add these connections, connect the input units, now you can learn associations between the features and properties. So not only can you learn that greenness predicts that it's a vegetable, you can learn that greenness predicts leafiness, or that leafiness predicts crunchiness, right? So now you are learning associations between features. And with this kind of model, you can actually fill in missing information. So if you have some of these features but not others, you can predict what the other features might be because of these correlations, right? So, one nice example of this is this kind of thing. So if you think of visual input, so can you read what is written there? Right? So you can all read that it says mind, right? But of course you can only read it because you are familiar with the language, right? And so you are using contextual information and you are basically doing a pattern association, right? The information you have in this picture doesn't tell you that this is an N, right? It could also be an H, for example. Similarly, some of the others, this could be a B, for example, right? But you are not reading it that. Without any effort, you immediately think that it is mind. So you are able to immediately fill in the missing information, and that probably is happening through some kind of association, because you have a frequent pattern in your mind and you see part of that pattern and immediately retrieve the rest of the pattern. Okay? So this is one powerful aspect of these kinds of neural networks, that they can fill in missing information. And another way of saying that sort of is that they are kind of robust to noisy inputs, right? So when some part of the input is missing or mutated or incorrect, you can possibly fill it in based on the part that you are actually observing. And this point is actually what we will see the relevant point for thinking about the original problem I was talking about, which is non-coding RL. Okay. So any thoughts at this point? Right. So I'll have to speed up a bit, but I'll just there's a bit of maths on these slides, but I'll try to describe it in kind of most high level way possible. So this is now I'm talking about the specific paper Deutsch 2016, I'll show you the reference at the end, which tries to use these kinds of auto-associative models which we have just described, neural networks which have associative connections, to try and model how these non-coding RNA like microRNA and so on might be in a distributed fashion regulating gene expression. Okay, so the basic model looks like this. So let's say you have a certain set of species of non-coding RNA. So by the way, this model is assuming that you have only non-coding RNA which regulate each other. But more generally, these species, some of them could be non-coding RNA, some of them could be coding RNA. Right? So the model extends to interactions between different kinds of RNA. Right? So let's just say you have n different types of RNA. They could be coding or non-coding. Each one has a certain concentration level. Right? So C1 to Cn is the concentration of the ith kind of RNA. And part of that is unbound. Right? So what we are assuming is that two different kinds of RNA can bind to each other, just like you saw in that picture where microRNA comes and attaches itself to mRNA. Right? So basically, RNA very easily are able to kind of bind to each other. Right? This is the difference between RNA and DNA. RNA is very reactive, and that is kind of why it is used as an intermediate step in by the cell. Right? So you have species 1, species 2, species 3, right? All of these are different types of RNA, and any two of these we are assuming can go and attach to each other, but the rate of attachment and the kind of strength of attachment, those things will vary. So the way we are going to model it is as a set of basically chemical reaction, right? So the ith and jth kinds of RNA can bind together to form a complex, which we are denoting SI-SJ. And just like in chemical reactions, normally you talk of the rate as being determined by the concentrations of this, so the rate this way will be determined by the concentration of Si and Sj. The rate this way is determined by the concentration of Si, Sj. So this is being modeled as a reversible reaction. So the equilibrium constant, you might remember from school chemistry, is given by this, right? So where rho ij is the concentration of Si, Sj, and rho I, rho j is the unbound concentration, as we have just said, of Si and Sj. So the free Si, Sj are available. So Kij is some parameter, it's some equilibrium constant which will depend on, let's say, the kinds of RNA that are there. Some of them will have a higher value, some will have a lower value. So, but if you imagine that you have values set for these, then you have a model of how these are interacting. And it turns out, again, we are not giving the derivation of this. If you look at this guy's papers, I think there's an older paper which actually derives this. But if you make these assumptions, at equilibrium, the relationship between the bound and unbound is given by this equation. Right? So this is the amount of the ith RNA which is not bound to anything, and this is the total amount. Right? So some equation, right? Again, here we don't need to worry about how this equation came, but you can derive. So this is the basic model. You have a bunch of different kinds of RNA which can interact, which can bind and unbind. They have equilibrium constants, and you can derive fairly straightforwardly what is the relation between the bound and unbound concentration. Right? Now the more interesting thing is on the next slide. Having set up that model, we now need to add on top of it some kind of model of the dynamics, which remember is sort of what this original picture was about. You know, if changes, how do changes in G1 and G2 affect G3? Right? So this picture was saying that if G1 goes up, G3 goes up. If G2 goes up, G3 goes down. So this is the kind of dynamic effect that we need a model for. So given that we have arrived at what we just saw, right? So the equilibrium level of rho I is given by this. So that means that to write a dynamic equation for rho I, you would need to have something like this. Right? So you see what is happening here. So the rate of production of rho I has a decay term which is proportional to itself. This is the normal first order kinetics, right? The more the concentration, the more the decay rate. And then the production rate has to be this, because only then will the equilibrium point be reached when this is equal to this. Right? So when rho I is equal to this, which is what the previous slide was, then this becomes 0. Right? And therefore the rate of change becomes 0 and you are now at equilibrium. So therefore it has to be this, and there will be some time constant here which determines the rate at which these association and disassociations happen. Right? So this is the rate of change of the bound RNA. So basically this is capturing how rapidly it binds and unbinds to other RNA. So you have this kind of equation. And now, so far we have just captured the binding. We don't have a model for the actual production of these RNA. So that now comes here. Right? And this is really what is trying to capture this kind of thing. So how is the concentration level of G3, for example? You can think of G3 and S3 as the same thing. Right? So imagine S1, S2, S3 are just G1, G2, G3. Right? So these are just three different kinds of genes or RNA. So to model the production of any of these, this is the simplest kind of differential equation model, which has, these kind of models are very widely used in gene regulatory network modeling in general. That the rate of change of CI, again there is a degradation rate, first order kinetics, it's just degrading at a rate proportional to the current concentration, that is standard. And the production is some function of the concentration of this and the unbound concentrations of all the genes. Right, so remember, the thing we have added here is that, so there are a bunch of S1, S2, S3 in the cell, right? But some of them are bound to each other. So when they are bound, they are ineffective, just like we saw with the microRNA. Right? So therefore, what matters is the unbound part, right? The part which is freely floating around and which can actually then go and activate something else, right? Just like transcription factors would go and activate genes. Right? So therefore, that is why in F you have there rho 1 to rho n, not C1 to Cn, right? Because it's the unbound part of G1 and G2 that can be affecting G3, or more generally of S1 to Sn that can be affecting. So this is some function. We have not yet defined what F is. That is coming just below. But some function of all the input genes or all the other genes is going to be controlling the rate of production, and the rate of degradation is going to be just a first order thing. And again, there will be some time constant which is a parameter to be determined. So the specific form of F, this is the key thing really, right? So you have this general regulatory model, but there is a specific form of F which actually leads to a model that captures associative memory. Right? And you can actually show, again we are not going into the full details, so you will not be able to show it in this talk, but you can show that by making a particular assumption of form of F, basically this model ends up being equivalent to the kind of model you saw a while ago, which was these associative models. So what is that form of F? So here it is. Again, if you have studied neural networks at all, you will recognize this as somewhat familiar. Right? So this F corresponds to, so there is a constant term here which is the proportion of the total concentration of the unbound, and then you have S. S is a sigmoid function. Right? So the simplest example of a sigmoid function is the logistic sigmoid. Sigmoid just means, the literal meaning of sigmoid is S-shaped, right, in Greek. So the thought of a sigmoid function is something which looks a bit like this, right? So it kind of has, so it's roughly speaking like a threshold function, a step function, but a smoothed version of that. Right? So the simplest example of a sigmoid is the logistic sigmoid, where basically Sx is equal to 1 upon 1 plus e to the minus x. Right? Just consider this function. So as you can see, when x is 0, this function will be 0.5. Right? So if you look at x equal to 0, Sx will be equal to 0.5. But as x goes away from 0, this will quite quickly go to the extremes. Right? So if x is, let's say, 10, then this quantity is very small. So basically this goes to 1 upon 1. Right? So very quickly in this direction it goes to 1. And in the other direction, if x is minus 10, then this is a huge number. So this has gone to almost 0. Right? So basically you get this kind of thing. And in order to sort of scale the sharpness of this, instead of x here you can put, let's say, beta x, where beta is a parameter greater than 0. Right? So if you want to make this even sharper, you put, let's say, beta equal to a big number, 100 or 1000. So then even a very small change in x will lead to a very sudden jump in this. So basically you can, this is a way of approximating a step function, but this is an actual continuous function which is better than a step function because you can use it to do analysis. Okay, so this S function is something like that. It's basically a step function, and in fact so in the neural network you saw that there is this threshold, isn't it? So this tau, so you are actually assuming a step function here, isn't it? Because you are saying that if less than tau you have 0, if greater than tau you have 1. So if you do the function that this is computing, it's actually a step function. And in practice, what is used rather than a step function is some kind of sigmoid function, because those are easier to work with, they are continuous. Right? So actually exactly this kind of function is used in those neural network models, and the same function is being used here. Right? And inside that function what do you have? So again, don't worry about the exact values, some arbitrary choices have been made, but the basic thought is that you have some kind of weighted combination. So look, this is a summation over all of the different unbound concentration. Right? So this J is basically going from 1 to n. So rho 1 to rho n is being taken and it's being multiplied by some constant 2, and then there's also dependence on the equilibrium constant. Right? So again, I'm trying to just present it in a very simplified way. There are a few things I'm leaving out, but the basic thought is that so think of it that inside these brackets, basically the same type of thing is happening as over here. Right? A bunch of inputs are being multiplied by weights, and you are taking the sum of those things. That is what is inside those brackets. And then On top of that, you are applying this kind of sigmoid function, which is basically doing the same thing as a threshold function. Right? So roughly speaking, do you see why this is similar to this? Right? Because you are taking a weighted combination of the different inputs and applying a kind of threshold to it. So it's something that is similar to this, and that's why the particular choice of this function ends up leading to a neural network-like model, which in fact turns out to be an associative memory kind of model. Okay, so this is basically all the maths I think we'll do. If you are interested, you should look at the paper here, because we have limited time, so I've just given a somewhat rough overview of things. So we are coming to the end. So this is again just a quick overview of the results that come out of this model. So they have also in the paper done some mathematical analysis of the model, which we have not looked into. So what I am showing here, so what ends up happening is that you set up this model and you set certain values for all these kij's, the equilibrium constants and so on, and it turns out that by setting particular values, this model can now effectively remember certain patterns. Right? And what is a pattern? It's just a setting of all of these values. Right? So unbound concentration of gene 1, gene 2, gene 3, or more generally non-coding RNA 1, 2, 3. Right? This model is for non-coding RNA, so it can be all kinds of RNA. So you have, let's say, 50 different kinds of RNA. In these examples, in fact, n is equal to 50. So 50 different kinds of RNA. Each one has some concentration. This is what we are calling a pattern. Right? So there is a certain set of concentration levels for all the genes, which is a pattern. And basically, if you set all the parameters in the model, it remembers a certain number of patterns, which means that those are the steady state solutions of the system, right, as I said. So of course it has to be a steady state, because the point is, when you set all these rows to these values, then all the derivatives become 0. Right? So basically all those derivatives become 0, so things stop changing and it's a steady state. So basically the steady state solutions to this model are the patterns that we are talking about. And the reason why it is like an associative memory is because if you now have some of these missing or messed up, right? So that means, of course, if you initialize the network with exactly this pattern, it will just stay because it's a steady state, nothing will happen. But supposing I tweak some of them, right? I randomly take 10% of these, 5 out of 50, and I just randomly tweak them. Then what happens? So that is what this picture is showing, right? On the x-axis, what fraction of those do you tweak, right? And on the y-axis, how often do you end up in a different steady state than the one that you started with? Right? So you start with this steady state, which is a pattern. 10% of the values you randomly just change to something else. And what happens? Then these different lines correspond to different settings for basically this beta parameter, right? So you don't worry about it, but the pattern is the same. So you can see that up to 10%, the fraction of mistakes is quite small, right? It's 10% or lower, and you can go up to 20-25% and not get too many mistakes. Right? So basically what is happening here is that kind of auto-association. Right? So out of all the 50 genes, if 10 to 20% of them have the wrong value, the system converges to the correct value. Right? So by observing some of the RNAs in the correct concentration, it automatically sets the other RNAs to the correct concentration most of the time, right? So, but if you increase the amount of noise, then obviously at some point, you start converging to other patterns. So this is showing that you can basically, just like in that mind example, you can deal with a certain amount of noise or kind of permuted data in the input and converge to the same output. And this other picture is showing you what happens. So left is changing the initial pattern, right is changing the values of the equilibrium constants, the Kij. So actually so the Kij's are the basic parameters in the model, right? So what are the steady states is determined by Kij. If I change Kij, it will change the steady states, right? So for a given setting of Kij's, I have a certain steady state, which is this one. Now supposing I initialize the system with this steady state, and now I just randomly change some of the Kij's, right? I set them to different values at random. What happens? So now the steady state of the system will change, but how much will it change? So one way to capture that is to look at something called the Hamming distance, right? Which you may have heard of. It's just a standard way of measuring distance between two strings, binary strings, let's say. Right? So anyway, for this purpose, all that matters is just some kind of distance measure. So how far away is the new pattern after changing some of these k's from the original pattern? And again, all this is showing is that you can actually make quite a large number of mutations, 50 or even up to 100, and get not a very large Hamming distance. Right? So the range of the Hamming distance is a 0 to 1. Right? 1 means that it's a completely different pattern, let's say. So value up to 0.2, 0.15 is not a huge change, right? It's a relatively small change. Most of the pattern is the same, even with very large number of mutations. Okay. So the reason why these are being called mutations, by the way, is that, as I said, the value of Kij depends on the exact sequence here, right? Because as we saw, the binding happens by complementarity, right? So if I change one of the nucleotides here, then the binding will become less strong or more strong, right? So one way to think of how these Kijs would change in this system is by mutations in the actual nucleotide sequence, right? Or the DNA sequence which is coding for these RNA. Right? So therefore, one way to think of this biologically is that you can have a lot of mutations in the non-coding regions, and you can still be pretty robust in terms of the patterns that you are producing of expressions of different genes and non-coding RNA. Right? So these two pictures basically are showing you that this model is in fact capturing the major properties of an auto-associative memory model, which is that you can retrieve the full pattern from part of the pattern, and that it is quite robust to changes in the parameters. Okay. Any questions?
So essentially this says that the system is pretty robust to changes that would happen in mRNAs, a large number of mRNAs.
Yes, non-coding RNA. So yeah, more generally non-coding RNA. It could be mRNA or this could be.
It's non-coding RNA.
Yeah. So, but it doesn't matter, right? So yeah, that is the thought. Okay. So this is the last kind of set of results from the paper. All I have left now is just to discuss, and this is some of the most interesting part. Unfortunately, I have run out of time, but I'll just quickly try and go through basic conclusions, both from the paper and then some of my own conclusions on top of that. Right. So the first few conclusions I've already mentioned. Model is pretty robust to mutations in the equilibrium constants, which can be thought of as caused by mutations in the RNA themselves. So this I have just said. Given a noisy pattern with up to 20 to 30% of the inputs altered, right, the different RNA concentration, the system tends to converge to the correct pattern. So this is the auto-associative aspect of the system. Multiple patterns can be stored. Right? Again, this depends on the settings of the equilibrium constants. If you look into the paper, there is some more analysis of how many patterns can be stored and so on, which I have not talked about here. So what this means is the network can respond appropriately to different environmental conditions. So depending on what the environmental conditions are, some of these RNA, let's say, might be more directly affected by the environmental conditions. So they will get set, and then they will drive the others. Right? So it can respond to changes in the environment by converging onto one of these steady states, whichever one is the appropriate one, because it is effectively acting as a memory for multiple patterns. Right? Then this general idea of distributed network computation, which is what this is doing, because as I have said, basically it is the same kind of model as a neural network model. It allows for robustness to mutation. So actually this is one of the general properties of neural networks which make them very useful. That because there are so many different features, so this is an example with two, right? But you saw that vegetable example, or you look at the typical kinds of examples, this face recognition on Facebook or whatever. Because you are looking at so many features, so if a few of those features are missing or wrong or noisy, it doesn't affect it too much, right? So you are actually quite robust to changes in the values of some of the input features, mutations, and in this case, as we have said, that is determined by sort of mutations in the RNA themselves. So in this biological system, what that implies is that you can have actually very high mutation rates. So if you remember that point on the first slide, right, where I was quoting this guy Deutsch himself, saying that non-coding RNA are thought to not be under evolutionary constraint, right, which means that they can kind of evolve freely based on what people are observing. They are not very strongly constrained by evolution because they don't seem to be very important. But he's saying maybe we need to revisit that idea, because even though they may in fact be under evolutionary constraint, that means they are actually important because they are not just junk DNA, they are actually regulating these genes. But because the way that regulation works is via this kind of distributed model, so there is a fair amount of flexibility, right? So actually if the concentration of the ncRNA changes by 10%, 20%, which means if the sequence of the RNA changes by a few nucleotides, the system will still be basically working the same way, right? That is what that picture was showing you. Okay, so therefore this is something interesting, right? So this is saying that maybe the way in which we decide that something is under evolutionary constraint needs to be rethought, right? And there may be certain things which are actually mutations are happening very fast, but that doesn't mean they are not important. It's just that there's a flexibility in the actual exact sequence that they can have. Right? So this is a very interesting point from the evolutionary biology perspective. Okay, so up to here is more or less what is there in the paper. Then a few points which I have added on top, which are kind of research ideas that I'm thinking about and would like to look at. So this paper as such doesn't really talk about learning, right? I told you about the perceptron, which actually sets all of these weights. Right here, all of the parameters in this model are actually physical constants, right? So corresponding to these weights and thresholds are the Kij parameters basically that are in this model, right? So they are determined by the actual sequences. So you might think there's no learning happening, these are just physical parameters. But how does the system come to be in a particular state, right? How do these sequences come to have particular values which cause these constants to be there? Of course, that is happening through evolution, right? So if there is learning happening here, that learning corresponds to the setting of these Kij values, these equilibrium constants, and that is happening through evolution because all these sequences have evolved over time to have certain sequences. So therefore, the thought here is that learning actually is happening via evolution, right? And there is some very interesting recent work from a different direction. Leslie Valiant is the key figure in this, who's a famous computer scientist, which actually tries to model evolution itself as a kind of learning, right? So I'm not talking about that here, but it's something I'm very interested in if you want to discuss later on. The idea that evolution is a kind of learning, and therefore you can think of, and that idea becomes especially interesting in this context because you are actually here talking about a neural network kind of model where the parameters are being set by evolution, and in actual neural networks the parameters are set by learning, perceptron model. So the evolution is doing the same thing here that learning does here, and more generally you can make this analogy between evolution and learning that is happening in this Valiant paper. Okay, so final thoughts. So the other thing is that this model doesn't have any intermediate representation, right? As I showed you for the XOR neural networks, powerful neural networks actually need intermediate representations, hidden layers. This model doesn't have, right? Because it's just allowing every non-coding RNA to interact with every other non-coding RNA. But what if we actually had a hierarchy of interaction, right? In the real biological system, it seems plausible because even in the first slide, I was telling you about these different kinds of layers of things that happen. So you have gene regulation, you have protein-protein interaction, signal transduction, metabolic pathways, right? So not everything in the cell is interacting with everything, right? There are actually some structure or hierarchy possibly to what is going on. And so if you want to go beyond what is done in this paper and say that rather than having everything interact with everything, and this paper of course is looking only at non-coding RNA, so it's looking at one level. If you want a more comprehensive model of the cell, which tries to look at everything inside the cell, maybe you want multiple levels, and then you actually end up with something like a sequence of representations rather than just one set of connections, and that could actually be something genuinely like a neural network. So neural networks with many intermediate representations of hidden layers have seen tremendous success in recent years under the name of deep learning. Okay. So one idea coming out of this talk is that could cells be doing something similar? Right? So this paper is already demonstrating that one plausible model for non-coding RNA is a neural network model which doesn't have hidden representation. But if you extend that to say actually to model the whole cell, you should have a hierarchy of representation. Then could you model the whole cell as a kind of multi-layer neural network, which would basically be a deep learning model? And how would the learning actually be happening in the deep learning model? So that deep learning model would have a lot of parameters and you would have to set those parameters by evolution, right? So the thought is that evolution and deep learning at some conceptual level are, they are related, right? Because both of them, why are both of these fascinating ideas? Which, so evolution of course is much older, but both of these ideas have one thing in common, which is that they made a huge splash kind of at the time when they came up. And the common reason for these ideas being exciting is that they are the most powerful mechanisms we know for the emergence of sophisticated adaptive and cognitive systems. Like starting from a simple kind of starting point, you can actually get something very complex which responds to a large number of different input conditions in different ways, which learns to produce complex functionality, right? So this is why these ideas are exciting, and evolution and deep learning are two of the mechanisms that we know about for that. So could they have something in common? This set of ideas is trying to give you some starting points for establishing such a connection, right? And I'm just finishing with this famous quote by Darwin, which is to back up this point about evolution, right? So this is the last line of Darwin's 1859 book, Origin of Species, right? Where he basically talks about why evolution is such a captivating idea, right? So life having been originally breathed into a few forms, and then whilst this planet has gone on cycling from so simple a beginning, so this is the key part, right? From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved, right? So the power of evolution is simple starting point, by this process of just a simple process of natural selection leads to tremendous complexity, tremendous adaptability, and one kind of actual mathematical computational model we have which can do something similar is deep learning, right? So therefore that's why I am suggesting that there could be something interesting in pursuing the ideas in this paper to try and flesh out that. Okay. So these are my references. This is the paper which I mostly talked about, the associative memory model for non-coding RNA. This is an archive paper, it's not yet published. This is the Valiant paper on evolvability, which tries to make the connection between evolution and learning. So this is an important part also of the broader conclusions. And this is the Steven Pinker book, How the Mind Works, where I took some of the discussion. Okay, thanks very much. Sorry I went a bit over time. So I've also given my email address, so of course if you have any questions right now, I'm happy to take them up. But if you found any of this interesting, would like to talk more, would like to discuss possible projects and so on, I'm always happy to do that. Feel free to write to me. Thanks.
So we'll have breaking reflection.